Good morning, everyone. Today we are going to discuss a few questions related to immunology. So let's start with the first question. A researcher observes defective neutrophil recruitment despite normal complement activation. Which molecular defect is most likely responsible? So let's discuss on this question. As we can see, there is a proper normal complement activation, but there is a defective neutrophil recruitment. So what is the most likely defect observed? So the answer is CXCL8 deficiency impairing CXC-mediated chemotaxis. Here, this IL interleukin 8, IL-8, is the primary neutrophil chemoattractant. So here, even with the intact complement, neutrophils fails to localize without chemokine gradients. So let's check this out. Okay, that was right. So coming to the next question, which complement fragment is most strongly associated with anaphylotoxin mediated master cell degranulation? So what might be the answer? We all know about the master cell degranulation, right? So which complement fragment is most strongly associated with anaphylotoxin mediated mast cell degranulation? So here the answer is C3A. C3A is an anaphylotoxin that triggers master cell degranulation, histamine release, increased vascular permeability, etc. So here the answer is C3A. So coming to the next question, a patient with recurrent intracellular mycobacterial infections most likely has impaired signaling through which pathway? So let's reread it. A patient with recurrent intracellular mycobacterial infection. Okay. What is the most likely impaired signaling through which this pathway? Which pathway? So here the answer is type 2 interferon receptor pathway. Let's go for this. So why is type 2 interferon factor? The IFN gamma signaling activates macrophages for intracellular killing via enhanced phagolysosome fusion, increased ROS regulated MHC expression, etc. So here type 2 interferon receptor pathway is which follows. Okay. So coming to the next question, which acute phase protein both activates the classical complement pathway and function as an opsonin? Okay. So what is the answer here? It is C-reactive protein. C-reactive protein is the acute phase protein. Both activates the classical complement pathway and function as an opsonin. Okay, CRP uniquely binds phosphocholine on on microbes, activates classical complement or antibody independent, and it also acts as an opsonin. Okay, it rises up to 1,000-fold within hours, which is better than ESR valuation. Okay. So let's go for this. Okay, next question is the failure of dendritic cells to home to lymph nodes would most strongly implicate dysfunction in which receptor? Okay, let's think it through. Let's reread. Failure of the dendritic cells to home to lymph nodes would most strongly implicate dysfunction in which receptor? So here we got the answer, which is CCR7. So CCR7, about that, it lead without CCR7, a failure to activate the T cells occur. Okay, if we can't activate the naive T cells, it lead to impaired adaptive immunity. So the CCR7 responds to CCL19 and CCL21, guiding dendritic cells to lymph nodes for antigen presentation. So without CCR7, what happens? It doesn't activate naive T cells, which lead to impaired adaptive immunity. Okay. Okay, let's go to the next question. Which defensin killing mechanism? Okay, defensin killing mechanism involved in what? Peptides inducing membrane curvature before pore formation. Okay, which defensin killing mechanism involves peptides inducing membrane curvature before pore formation? Okay, the answer for this is the toroidal model. Okay, toroidal model in this pore mechanism, and the peptides insert into the membrane, then lipids bend inward continuously, and then the pore lined by both peptides and lipids. So the defensive killing mechanism which involves peptides inducing membrane curvature before pore formation is toroidal model, and this is most commonly tested. Okay, let's go for the answer. Okay, a neonate presents with recurrent infections but normal adaptive immunity. Recurrent infections is present, but normal adaptive immunity is also present. So deficiency of which molecule is most likely occurring? So here the answer is mannose binding lectin. MBL is crucial because neonates have immature adaptive immunity. Here it functions in cases of pattern recognition, complement activation, or lectin pathway, and opsonization. So if there is a deficiency, recurrent childhood infections occur in case of neonates. Okay, even though there is a normal adaptive immunity. So let's go for the answer. So bear in mind infant plus normal antibodies, then think about MBL. Okay. So going for the next question, which cytokine is the master regulator of hepatic acute phase protein synthesis? So we are talking about the cytokine, and we have to know which is the master regulator of hepatic acute phase proteins. Okay, which one of the hepatic acute phase protein is the master regulator? Okay, we have to know that, and which one is that? IL-6. Okay, here, as we all know, as the master regulator of hepatic acute phase synthesis triggers production of CRP, fibrinogen, serum amyloid A, ferritin, etc. It is produced by macrophages and endothelial cells during inflammation. Okay, what produces IL-6? It is produced by macrophages and endothelial cells during inflammation. It's just a high yield chain. That is, for example, in case of an injury, IL-6 occurs, then goes to liver, and APP surge, acute phase protein surge is present. Okay. Okay, now going to the next question. Which chemokine is primarily responsible for organizing B cell follicles within a secondary lymphoid organs? Let's reread the question. Which chemokine, okay, is primarily responsible for what? Organizing B cell follicles within what? Within the secondary lymphoid organs. So you're going to talk about the B cells follicles within the secondary lymphoid organs. Okay, so we want to know about which chemokine is primarily responsible for organizing these B cells in the secondary lymphoid organs. So the answer is CXCL13. So in case of CXCL13, which organizes the B cell follicles in the secondary lymphoid organs, without the presence of this, the germinal centers fail, antibody affinity maturation suffers, etc. This is highly critical for the immune microanatomy. So remember, CXCL13 is equal to B cell zip code. So coming to the next question, low haptoglobin combined with elevated lactate dehydrogenase most strongly indicates which pathophysiological process. So question repeating. Low haptoglobin, okay, it is combined with elevated lactate dehydrogenase. Okay, now haptoglobin is low and lactate dehydrogenase is high. And what is this pathophysiological process involved here? What is it? It is intravascular hemolysis. Okay, intravascular hemolysis is the condition where the haptoglobin binds free hemoglobin. Okay, and when the RBC destruction occurs, hemoglobin is released into the plasma. Haptoglobin is consumed, which leading to the levels dropping the levels of the haptoglobin, and LDH rises from the cell breakdown. Thus, haptoglobin level decreases, and the LDH level rises from the cell breakdown. So the classic hemolysis triad is happening here. That is, haptoglobin is decreased, increasing the LDH, increasing the indirect bilirubin. So we have covered about chemokine versus complement roles, cytokine hierarchy, liver immune axis, membrane microbiology, and neonatal immunology, and biomarker interpretation, etc. And we have to recall all the things related to this. And thank you for your time. And let's go for the next quiz to attempt the ultra advanced soluble factors grand quiz. Okay. Hope to see you on the next video. Stay tuned.